We're going to continue our study of the respiratory system by doing a comparative look at how respiration occurs in, we're going to start with fish and look at gills and then formation of the air sacs, the air bladder, and so forth. So in our earliest example, in the fish, we're going to be looking at the lamprey. So let's try to draw kind of what goes on in a lamprey to understand how its respiratory system works. So anyway, in a lamprey, I've only put 5 pairs of gill chambers on here instead of 7, but water would come in typically through the oral cavity here and move backward through the pharynx. And there's a place here at the back of the pharynx where we separate the water flow from the food flow. So food would go into the esophagus, water goes into a branchial tube. So there's an opening to a branchial tube here, and the rest of this would be the branchial tube in the anatomy of a lamprey. And along the wall of that branchial tube are openings, little pores that allow the water to move from the branchial tube into the gill chambers. So we have a number of gill chambers here. And inside the gill chamber, we have our gill plates. And on the surface of the gill plates, we have what are called gill lamellae. Higher fish have what we usually call gill filaments. These fish have gill lamellae, as do the sharks, also have the gill lamellae. What a lamella is, as opposed to a gill filament, let's see if we can do this. It's on the surface of the gill plate, instead of having a long pointy structure sticking up, filaments sticking up, what we have are these low ridges, very thin, fleshy ridges that stick up in a row or a series along the surface. So these are arranged so the blood goes in, circulates through each of these lamellae, each of these layers. So you have, it kind of looks like a bunch of feathers side by side. Anyway, thin plates that are aligned in a series on the surface of each of the gill plates. And we call those gill lamellae. These are the gill lamellae right here on the surface of a gill plate. So typically the water comes in here and just moves through the gill chambers and out of the excurrent openings, the external gill openings on the lamprey. Now there is a way for them to respire if their mouth is busy, if they're feeding at the time and they're not allowing water to come in there, they can pump water into and out of the gill chambers from the outside. So it can come in and out and in and out and in and out on the outside through the external gill opening out here. So lamprey and hagfish do this basically the same way. In the hagfish, there are some species where the excurrent opening combines with the next excurrent opening and so on. They kind of form a tube that has a common opening further back, so they have an extended tubular structure that accepts water from each of the gill chambers and then exits further back. Depends on the species, but anyway, there's some variation in how that excurrent tube is arranged in the hagfish. Lamprey all do it basically the way it is here on the board. Next is the shark. In my drawing of the shark here, this represents the opening of the mouth about right here. If we cut through the pharynx and look at the roof of the pharynx from below, we would see these two spherical openings. Here's a spherical here and one on the other side. Those are also openings that allow water to move into the mouth and pharynx. So water comes in the mouth and water comes through the spherical, two different openings. So that's kind of an inhale for a shark, bringing the water in. And then the water moves back and through these gill chambers to the outside. In the typical shark, most sharks, they have 5 gill openings on the side, meaning that they have 1, 2, 3, 4, 5 gill chambers with respiratory surfaces in each one. Some sharks are on the move all the time and are always having water to move through their pharynx just by having their mouth open while they swim. That's how they get most of the water to move through to give them the oxygen that they need in their musculature and so on. Some species of skates and stingrays that lay on the bottom, they bring most of their water in through the spherical, and you see the spherical open and close and open and close and open and close as they pump water through their pharynx. We're going to look now in detail at a single gill structure in the shark. I'm going to draw that next. So I'm not sure why I drew this with a kind of a bow in it, and I kind of drew this a little bit large, but we can still label the parts. In blue, outlined in blue in here, is the cartilaginous component inside of each of the gill structures. So this is part of the gill arch. Let me put that in blue. Gill arch, that's the skeletal component in the shark. It's made out of cartilage. To the inside of the pharynx, there are these extensions that stick out here that are kind of like blunt teeth that stick out, just little pointed things covered with soft flesh, but supported by this internal skeletal component. These are called gill rakers. So there's a gill raker sticking out there. There's an extension of, there are several extensions of fine cartilage that reach out into the gill septum that divides the two sides of the gill out here, and these are referred to as gill rays. So this is a gill ray. Those are the cartilaginous components you see in the gill out here. The arch component, the raker, and the ray. In this area around the gill arch, the cartilaginous component there, there's also a blood supply coming in, an artery branch, at V1 on either side over here, and an incoming artery and an outgoing artery, so I can't really compare them very well. And there's also, excuse me, a nerve found in there. This will represent my nerve. Later on, when we look at the nervous system in humans and other mammals and so forth, we're going to talk about having the 5th cranial nerve going to one arch, the 7th cranial nerve going to another arch, the 9th going to another arch, the 10th going to another arch. So cranial nerve numbers are unique to a certain gill arch or pharyngeal arch structure out there. So there's a nerve component, there's a blood supply in that region. Now some of the blood, of course, is going to flow out into the gill over here. This part out here between the red gill lamellae in there is just referred to as the gill septum. There, and these are the gill lamellae. Now on a shark, that gill septum goes all the way out, and on the outside of the body, there's a bit of a plate of the gill septum on the outside, the exterior plate that divides one gill from the next out there. Now, with water flow coming through right here in front of this gill structure, and back here on the back side also coming through here, they name this front section to the front of the gill the post-traumatic demibranch. The post-traumatic demibranch. Let me write that in there. Post-traumatic demibranch. That means it's behind the water flow. Down here, this one, these are in front of the water flow, so this is a pre-traumatic demibranch down here. So that naming makes things a little bit confusing because post you would think would be on the back side of the gill, but no, it's on the back side of the water flow. And pre-traumatic is to the front of the water flow. That term traumatic refers to the turbulence or disturbance caused by the water, the flow location. So this is in front of the flow, this is behind the flow of water through the gill. A demibranch is one side of the gill structure. The respiratory surface on one side of that gill septum that divides the two surfaces. Here we're going to do a quick comparison between shark ratfish and a teleost fish, a higher bony fish. This is what we just looked at with a complete septum dividing the pre-traumatic and post-traumatic demibranch sides of the gill, and it reaches out and there's a plate out here that divides one gill opening from the next. In the ratfish, remember, it has an operculum, so it has a cover over all the gills. It has a shorter septum. It still has a septum, but it's shorter, and off of that we have the gill components, the respiratory components, come out and extend on beyond that gill septum a bit within the gill chamber. This is exaggerated even further in a teleost fish, so I'm going to label this. This was shark. This is ratfish, also a cartilaginous fish, right? That holocephalin with an operculum. And then most of our modern bony fish have this arrangement where they have the post-traumatic demibranch set of gill filaments coming out, and then the pre-traumatic set coming out. So it looks like they have two gills for every arch when you look in their gill chamber, when you look under the operculum of like a bass or something like that. When you look in there and look at their gills, it looks like there are more gills than what you would expect for the number of gill arches that are in there. So anyway, you have this one set and then the second set coming off of each gill arch in a bony fish. And in the bony fish, instead of having the lamellae, these are now called gill filaments. Because instead of being simple plate-like things with a flat edge on it, they look something like this. Each of these surfaces. So there are a number of filaments rather than just being a flat-topped plate there. And then there'd be another series and another series and another series of filaments in the gill, as we see here. We're going to talk for just a minute about external gills. We've talked about some rather normal kinds of gills, those respiratory surfaces. And I've just drawn one side of the head of the animal here. We see this in some like frog tadpoles and in some larval fish. So this is mainly a larval characteristic I'm drawing here, but if the respiratory surface that occurs along a gill produces long extensions of the gill filaments that go out into the water. This is one kind of external gill coming out here. So they have their origin inside under an operculum, but extend out beyond that opercular covering, some kind of covering of the gill, and extend out into the water beyond the gill chamber itself. So that's one kind of external gill structure. The other kind of external gill structure, there are actually structures that grow from the outside of the body, from the outside of the gill structure, out beyond here, and on that structure, you get gill filaments to develop out there on the outside. So you get a number of gill filaments off of each of these projections that go out into the water here. I've drawn 3. In some species there's only 1, so it varies according to what we're talking about. But salamanders do this, and if you're familiar with mud puppies, they have large feathery gills on the outside of their head. It's common in sirens, if you know what a siren is. Also, they would show that. Lungfish, some of the different species of lungfish have an external gill structure in their early development, and some of them keep it until they're pretty good sized fish, in addition to what gills they have to the inside. And of course they're lungfish, so they have lungs as well. Polypterus does this as a larval, well not larval, but a young Polypterus up to a few inches long, possibly, will show a nice external gill structure here. All larval salamanders, if they go through a larval stage, an aquatic stage, they have external gills that are of this type. So they actually grow an external projection out here coming off the outside, and then on that they have the gill filaments. So that's, like I say, some primitive fish, lungfish, Polypterus, are 2 examples of that. Salamanders, most salamanders have a larval stage where they have external gills of this sort. This present in some teleost fish as larval fish, and some tadpoles, frog larvae as well. So anyway, 2 different kinds of external gills. Okay, we're going to talk about 2 kinds of what we consider lungfish right now. Some people consider Polypterus a lungfish for reasons we'll see in a minute, and then what we usually refer to as a lungfish as well. So in Polypterus, first of all, off this green tube I've drawn in there on both of these represents their digestive tube, their gut. So there's a pharynx up here in the front, and then esophagus back to the back, and somewhere near where that pharynx reaches the esophagus, we get the development of a tube that will lead into the air sacs. And so this branches right away into 2 different branches in the Polypterus, and so we get an air sac on one side. On its left side, it's a little bit smaller than the lungs that develops over on the right side. So I've got another one developing over on the other side that's a bit bigger. So they're unequal. Small left and a large right air sac developing there. On a lungfish, they both go off to one side. So you've got exit here, and then you have one and then a second air sac developing off of one tube. And they're both over on the one side, on the right side of the lungfish. So they both have 2 lungs, 2 air sacs, or 2 air bladders, depending on how you want to call them. But this is more typical to have a left and a right, but they're not equal in size. And then in the lungfish, they're both pushed to one side of the body, and again, they're not equal in size. So this is the lungfish condition, this is the Polypterus condition. To look at it in a cross section here, let's see if we can do this. If If this is the gut, right here in green, off the bottom of that gut is where we have the connection to the air sacs. So in a polypterus, they kind of form one on one side, and then they form a larger one on the other side, like that. So this is like a very short kind of trachea that splits into two primary bronchi that go into those air sacs. In the lungfish, here's gut. And you've got two air sacs on one side of the body and a lungfish. In the higher fish, higher bony fish, you see some variation in the arrangement of things. The green here represents the gut in each of these pictures, these three pictures. Sometimes the attachment of this, and these just all have a single air bladder, not the double air bladder that we saw in Polypterus and the lungfish. But anyway, sometimes that comes off the ventral side, like it does in a Polypterus or lungfish. So it can be ventral, but sometimes it comes off the side of the gut, and sometimes it comes off the top of the gut, depending on the group of fish you're talking about. So in these cases where there's still an attachment to the gut, these fish sometimes have the ability to go and gulp some air from the surface of the water and essentially push it into their air sac to add air to their air chamber here. Fish that have their air chamber attached to their gut are referred to as physostomas. Stome referring to an opening. There are a number of fish groups that have the air bladder develop initially off of the gut, but in the adult fish and the final anatomy of the fish, there's no connection between the gut and the air bladder. These are referred to as physoclistus. Now, most of these fish use their air bladder primarily as a buoyancy device. An air bladder can be used for buoyancy. It can be used for respiration, as it is in many examples. So buoyancy, respiration. It can also be used to either produce sound, amplify sound, if it's a kind of fish that makes a noise in the water. Sometimes they use their air bladder as kind of an amplification system. And it can be used to amplify sound, to receive sound, for hearing, to receive vibrations in the water. So it has those different functions, the air bladder. But if you have an air bladder that's separate from your gut, or even in some of these, they aren't going to be always going to the surface to get air, but they're going to use it as a buoyancy device. And the way they control the buoyancy device is through a structure called a red gland. We'll just put a little part of it right here and label it red gland. Basically what it is a set of capillaries that concentrates the blood gases in one location near the surface of that chamber. So by controlling blood flow to that red gland, they can control what volume of blood gases are added to that chamber. And then other blood vessels, capillaries elsewhere along the wall of that air bladder, can remove blood gases, or the gases from that chamber, to add it to the blood. So you have both the contribution and removal of gas from that air bladder, very simple mechanism of controlling blood flow to two different locations. And if you watch a fish in a pond or an aquarium, you can just watch them. Many of the species have the ability to just rise up when they want to and then drop down as they want to. Their buoyancy is completely controlled by this vascular system involving a red gland for depositing or delivering blood gases into that space, and then other capillaries for removing that and reducing the blood flow to the red gland. So that's how we can control the volume of air in an air bladder in a physoclistus fish, and some of these fish do that as well. Now we're going to look at the lungs of different kinds of animals, but the nature of the lung membrane, of the wall of the lung, what that's like, and some of the added elements to the lung that we see in the different taxa as we go through here. And after we do that, we're going to talk about ventilation of the lung, how the body uses muscular groups, different muscular groups and different taxa to move air into and out of the lungs. In the fish that we've talked about, for the most part, the air sac is a very simple membranous lung. Its wall is really thin. You can see right through it. We'll let light pass right on through. It's very thin, transparent kind of lung. We see that in frogs and salamanders as well. So anyway, that very thin membranous lung, we see it in the fish and in the amphibians. A few of the amphibians and in the lungfish, we see a lung that has septa along the wall. So if you had a single lung coming out here like this, you would see along the wall that it is thrown into minor folds, and it's described as being honeycomb-like in the lungfish. But anyway, the wall would have these low septa that kind of increase the surface area across which you can absorb your oxygen. So that's the main concern with lungs, is getting as much oxygen as you can. The higher the oxygen demand of the creature, then the more surface area you need for absorbing that oxygen. So like I say, in some amphibians and in lungfish, you see these low septa dividing up on the wall of the lung. Like I say, it looks a little like a honeycomb on the wall of the lungfish. In a reptile, instead of having that low septa, we in some species have something more like this among the reptiles. But in most reptiles, we have a spongy wall, so that if you were to cut through it, you would see that the tissues make deep folds in it that form these cavities that they're actually more organized than what I've drawn here. But anyway, their surface area is increased over what you see in the example of the lungfish and some of the amphibians here. So reptiles have a spongy wall. Some amphibians and lungfish have septa, these low partitions on the wall, and then most fish and amphibians have a membranous type of lung. So this only leaves for us to consider beyond this, birds and mammals. So that's what we'll look at next. In a bird lung, we have a fairly complicated respiratory system, more complicated than what you see in mammals by some estimations. But anyway, let me do this in a different color. Do it blue. This would be incoming air. It comes in through the main primary bronchus, then enters a tube that's going to branch out and lead into these air sacs off of the main body of the lung. So we have all these tubes that lead into these air sacs back here. So incoming air is brought into these air sacs. So this is our lung right here, and these are air sacs. Supplementary to the lung, and in addition to that. These air spaces of the air sacs, some of them also have extensions that go out into some of the bones. You've heard of the bones of birds can have air spaces inside it, and then they would stem from these air sacs that are out here. And this is just one side, and you have the same thing on the other side of the bird. But anyway, so incoming air, when a bird inhales, it's bringing air into this space here. Then it will exhale, and in exhaling, let me put it in green, we have the air to exit these spaces and not go back through this main blue chamber here, but through a whole bunch of small tubes that are going to be leading back out of the lung. Those spaces in there, if you look inside the lung, you will see that there are cavities, and around those cavities, there's that spongy wall arrangement. So that there are air spaces, little cavities running all through the wall around this tube. And it'll take me a minute to kind of draw this in here. But anyway, lots of pores leading into a spongy network that surrounds this single tube. And this is just one of several tubes we have out here in the wall of the lung, in the sides of the lung. So anyway, this is a single parabronchus. Parabronchus. So the lung will have a lot of these, and the airflow going through here is in one direction. So unlike mammals who mix the air that they bring in with residual air that was still in the lungs when they exhaled, the birds are always pushing fresh air through these parabronchi, delivering more highly oxygenated air to those surfaces to absorb oxygen out of the air in the lung. So in the next inhalation, it's going to bring more air in and then push the remainder of that air out in the second exhalation. So the first exhalation brings it into the lung from the air sacs, and the second pushes it on out. So it's kind of a two-cycle thing. It comes into the air sacs, goes into the lung, then goes out from there. So parabronchi are the vital part of the lung in a bird. We assure that one-way flow of air out through these parabronchi from the air sacs that surround it. I haven't done all that great a job of trying to draw this mammalian lung, but anyway, this would be part of the mammalian lung, looking at this here. So what we have is a bunch of branching tubes. It comes in as a primary bronchus, and it branches into secondary bronchi. And those branch again into tertiary bronchi. And then we eventually have bronchioles. The bronchioles continue to branch into smaller and smaller bronchioles. Eventually you get what's called a terminal bronchiole. Terminal bronchioles lead into what we call respiratory bronchioles. Which lead into alveolar ducts. This is an alveolar duct here. Alveolar duct. And the alveolar duct is surrounded by these little pouches. These are the alveoli. Single one is an alveolus. So we've been over that with the human respiratory system before already. So we kind of knew all of that, but all mammals have basically that same pattern of respiratory branches in their lung. Now we're going to talk about ventilation mechanisms for different kinds of vertebrates. If you're a fish that has a lung, like a lungfish or a polypterid, that's going to take air into your pharynx, you can just kind of push it with the wall of your pharynx, push that air back into the air sac there. It's not, it's kind of like, almost like swallowing, you just push it back there with the pharyngeal wall. So that's what fishes do, which can use their air sac in that way. In amphibians, like a frog, the example we have here, they use primarily their throat region to ventilate their oral cavity and lung together. So the way this works, this is a two-step process in a frog here. So in this first example up here, let's use blue for the air. The frog lowers its throat right here with its nostril open. So here the nostril is open and allows air to come in as a volume into its oral cavity. So this is open nostril right here, and throat comes down. So that's the first event. You pull air into the oral cavity. Next, you close the nostril. Nostril closed here, and your throat goes up here instead of down. This was down. Now it's going to go back up. And when it goes up with the nostril closed, instead of pushing the air back out the nostril, it's going to push that air volume back here into the lung. So we get our air volume into the lung by pulling the throat down with the nostril open, closing the nostril, pushing the throat up. It pushes that air back into the lung. Now we need to get that air back out at some point. So with our nostril closed, we lower the throat again. So here's our throat back down. Again, it's going to pull that air out of the lung and into the oral cavity. And then the next step would be to push it out of the oral cavity through the nostril. Go in this direction. So here we have, this goes back up, nostril open. Now, I've watched frogs before, and they don't alternate very well with the closing of their nostril between the up and down movements of their throat. So they've got their throat moving at a pretty good clip, and they'll just occasionally close their nostril and occasionally open their nostril. But it has the basic effect of doing this kind of two-stroke arrangement with ventilating the lungs. In the mouth, then push it back to the lungs, then draw it out of the lungs, and then push it out of the mouth. So that's how amphibians ventilate their lungs. There are a number of salamanders called lungless salamanders that don't have a lung. They just ventilate their oral cavity and breathe through their skin. A lot of frogs do. Frogs have a lot of moist skin also across which they can get oxygen into their bloodstream. So they have both cutaneous respiration on their skin, and they use this oral cavity space. You can watch their little throats moving up and down. So that's amphibians. Reptiles, well, there's different kinds of reptiles, but to think about what they're doing, they have their rib cage. Frogs and salamanders don't use a rib cage to ventilate their lungs. They just use their throat muscles below their mouth. So in reptiles, you have this developed rib cage in most crocodilians, lizards, snakes, and they expand their ribs with their external intercostals and then contract the rib cage with the internal intercostals. So that's how most lizards and snakes do their breathing, is just with their rib cage, to expand and contract their rib cage with intercostals. Now we need to describe something special in the crocodilians, a special way that they can assist their rib cage in the ventilation of their lungs. Okay, a crocodilian can do something like diaphragmatic breathing. It doesn't have a diaphragm like a mammal has, but it has a structure that separates the lung from the rest of the abdominal pelvic cavity back here, and this is called an oblique septum. So you've got this partition, that wall of membrane that divides the lung space from the rest of the space back here. A nearly complete isolation of the lungs in their own cavities up there, which was not present in the most of the other reptiles. Some lizards have this kind of arrangement, but the amphibians don't. The lung is not divided from the rest of the abdominal pelvic cavity in there. So this is liver right here. The liver's kind of tucked up in between those oblique septa that isolate the lungs. Attached to the membranes and connective tissues around the liver, we have a special muscle. I had to put it in green because I kind of ran out of color, other colors to use. But anyway, this is called a diaphragmaticus muscle. It attaches way back here at the pelvic girdle. So there's the pelvic girdle. When it contracts, it pulls back on the liver, and these structures around the liver are pushed back, and everything is pulled back here next to the oblique septum. So that oblique septum falls back, comes back, and that allows greater inflation of the lungs. Now they have the rib cage as well working for them, so they can expand their rib cage and they can pull their liver and organs back behind the oblique septum to work it like a diaphragm, similar to the way our diaphragm helps us to breathe by moving our organs back down in the abdominal cavity, increasing the space for the floor of the lung. But anyway, this is the diaphragmaticus muscle. Birds do something similar to this. They have kind of a ligament in there that joins two structures behind an oblique septum, so that they can kind of make movements of their body that pulls back on their oblique septum a bit to help them breathe. It's not the same as a diaphragmaticus muscle in a crocodilian, but it works on the same kind of principle in there. So anyway, it's kind of a unique situation in crocodilians. A couple of other comments before we leave respiratory system. I failed to mention when we were talking about reptile lungs that some of the lizards have accessory air sacs associated with their lungs, very similar to what we saw in birds. But it's also thought to be true in dinosaurs that they had accessory air sacs associated with their lung structures. So it's not unique to birds to have those accessory air sacs off of the lung. In some of the reptiles, they use those accessory air sacs, the lizards that we're aware of, they can use those accessory air sacs just to inflate their body to make them look larger, to kind of do a threat display against intruders, enemies, in territorial disputes with other males of the same species, and so on. So they have that ability. It's known that the chuckwalla lizard down in the desert southwest of the United States can get in between rocks and kind of inflate its body when it gets into a tight place and make it very difficult to remove that animal from it wedges itself in there, so it gets more secure and protected against predation that way. Also, something about birds. I mentioned how the ventilation pattern works in birds, and that they can use a diaphragmaticus-like movement, not a muscle, but a movement of the body to pull the oblique septum back. But they primarily use their rib cage, and in flight, that flight movement ventilates their lungs, pushes the air in and out through the air sacs and through the parabronchi of the bird lung in there as well. So those are a couple of other things. We didn't talk specifically about mammalian respiratory movements here, but we did when we talked about the human respiration, and all mammals are basically the same. We have the external and internal intercostals that can expand and contract the rib cage. We have the diaphragm muscle, which is unique to the mammals, that pulls back on the floor of the thoracic cavity to inflate the lungs that way. So most of these use a negative pressure to inflate the lungs, to pull the air into the lungs. They just expand the space of the pleural cavities to do that in one way or another, either by an oblique septum, a diaphragm, the rib cage expanding, or something of that sort. So the frog was a little bit different in using a push mechanism, pushing the air back into the lung. So anyway, that's the respiratory system.